Okay, let's kick off. Welcome everyone. This is the 5th public seminar of the Q-Mare project seminar, where every month we invite an expert to help explore the complexity of natural and human-induced changes in marine ecosystems over time. So before we start today's seminar, just a wee bit of information just to help us on our way. So this is a 1-hour seminar. At the end of the hour, please feel free to leave, or you can stay if the speaker would like to continue the discussions. At the bottom of the Zoom window, you can see options for ways of interacting with the speaker and the community and asking questions. You can write your questions in the Q&A button, and we'll get to them in order at the end of the seminar, or you can wait until the end and then raise your hand when the speaker asks for questions. And I'll invite you to speak, and you can ask your question directly to the speaker, or you could write them to me and I will ask them for you. Also, you can use the chat button at any time during the seminar to share something of interest, links or anything that the community might like, something positive about the presentation. Okay, so let's jump straight in with that. It's my absolute pleasure to introduce our 5th Q-Mare seminar presenter, Dr. Jan Steger. So Jan is a postdoctoral researcher at the University of Vienna in Austria, and he's been doing some really fascinating research into the historical context of one of the, probably the most important human-driven biogeographical events in the world, and that is the invasion of the Red Sea fauna into the Mediterranean through the Suez Canal. And Jan has been exploring the functional and ecological consequences of this invasion using recent fossil records and all sorts of other approaches. And he's been uncovering some really interesting and sometimes kind of counterintuitive results, which bring a much needed context to understand this invasion and make future predictions. So it's absolutely perfect for a Q-Mare seminar series. So thank you, Jan, for giving us this presentation, which is entitled Patterns of Ecological Impacts of the Lessepsian Invasion, Novel Insights from Molluscan Living and Death Assemblages. So go ahead, Jan, please share your screen and take it over. Thanks so much.
So, okay, thank you very much, Aaron, for the nice introduction. I hope you can all see the screen and hear me well. As already said, sorry?
Looks good, Jan. Go for it.
Okay, great. Thanks. So I'd like to present to you some of the work we've been doing here at the University of Vienna on the so-called Lessepsian bioinvasion, the largest marine biological invasion, and also show you a bit how the combination of approaches from paleontology and modern ecology can help to understand this invasion in particular, but also other invasions in general. As you all know, the world's biota is changing at an increasingly rapid rate. This is related to all the different facets of global change, with the most prominently featured being usually climate warming, which is also very much mentioned today in the news. But there's also another, probably equally worrying process going on, which is the continuous erosion of natural and long-standing biogeographic patterns. And this is related due to human transportation as a result of economic activities, travel, tourism, and the like, which connects very remote places on the globe in a matter of days or even hours sometimes. And as you can see here, just as an example, the global shipping routes placed on a map, there's really a dense network of connections all over the globe. And it's clear that from time to time, despite measures being taken, organisms will be unintentionally transported by these actions to remote places. In the worst case, the organisms may establish self-sustaining and permanent populations, and sometimes even spread in the recipient ecosystem, which may have detrimental effects on the local ecosystem, the fauna, biodiversity, which is prominently known from the Hawaiian Islands, where a carnivorous land snail was actually deliberately introduced, and instead of eating another invasive snail as an agent of biocontrol, It actually preferred the endemic native land snail fauna that was driven almost to full extinction. On the other hand, similar invasions are known from the marine system, such as the lionfish that was introduced to Caribbean coral reefs, where it's a voracious predator feeding on all kinds of fish that can fit into its mouth. But apart from the direct ecological consequences, invasions may even have economic impacts, for example, on fisheries, or they may affect human health directly. For example, in the case of toxic species such as the Red Sea pufferfish that is now also appearing in the Eastern Mediterranean, or because non-indigenous species may serve as a vector for parasites or diseases of all kinds. So because of all this, today the invasions are recognized as one of the major threats to global biodiversity. However, this was not always the case, although the phenomenon is much older and it has occurred for at least centuries, some even say millennia. But of course, it's very difficult to find out about such early introductions because they may mistakenly be classified as native species. This was, for example, the case for the soft-shell clam Mya arenaria, and only the fossil record revealed that it actually was introduced by man to the northern European shores. Or the case of the oyster Chrysostria angulata, which was even termed the Portuguese oyster in its common name, but in fact originated from Asia as revealed by genetic evidence. Of course, invasions are not just an independent facet of global change, but they also interact with many other drivers such as habitat destruction, pollution, but also very prominently climate change. Climate warming is in fact facilitating invasions in two different ways. Several of the species that are introduced and now rapidly spreading are known to be thermophilic with respect to the climate of the system they are introduced to. And on the other hand, warming or climate change in general, such as changing precipitation patterns and the like, may also put at selective disadvantage the native biota, which have evolutionarily adapted to the long-term climate or average climate conditions. Whereas any propagules that are brought into the system may actually be directly selected for the new altered conditions and therefore have a selective advantage. Our system, as mentioned already, is the Eastern Mediterranean Sea. This is a semi-enclosed basin, which is highly interesting for ecological purposes because it's warming at a very fast rate, much faster than the global average. And on the other hand, it hosts more non-indigenous species of tropical origin than any other marine region of similar size. The reason for this is here on the lower right of the map, the Suez Canal, which is a man-made waterway with fully marine conditions that connects directly the northern Red Sea with the Eastern Mediterranean Sea. It was opened approximately 150 years ago and started off as a rather narrow and shallow canal, which was manually excavated more or less with very simple means. But over time, as vessels grew larger and the shipping became denser, the canal was enlarged several times to fit the actual demand. So today it's more than 20 meters deep and a few hundred meters wide. So the volume of water that can exchange between the northern Red Sea and the Eastern Mediterranean has increased considerably. This is reflected also by the number of non-indigenous species in the Eastern Mediterranean. And you can see here an example for Israel, which is quite representative for the whole southeastern Mediterranean Sea, where already several hundreds of non-indigenous species of Indo-Pacific origin have been introduced, and there is no end visible. So the rates may even have accelerated, although it's difficult to judge because we often face significant time lags in detection of non-indigenous species, because it's not always easy to find them, especially when they're small. What is clear, however, is that many of those species are quite rapidly expanding within the Mediterranean Sea. And when we consider here on the lower left, the different phyla that have been reported, the mollusks are by far the most diverse one. And because of their ecological importance and diversity, they are also an incredibly interesting study system, which we focus on in our working group. To give you an example of what the situation actually looks like, let's have a look at the sample that we took in the project. This one here is from Israel, from approximately 20 meters water depth on a soft bottom. And now let's mark in color, in red, for example, all the individuals that belong to Lessepsian species, and in green color, all those that represent native individuals. And the outcome is really obvious. So more than 90% in this case, and this is not an exception, are actually represented by Indo-Pacific species. So there's a major dominance of those in today's assemblages in the area. On the other hand, equally compelling, there are very few native individuals present and very few native species, which is quite worrying, and it in fact represents the most massive loss of marine biodiversity that has been documented to date. When we look a bit closer, quantifying this based on the shallow subtidal, which is the most affected depth range between here approximately 10 and 40 meters on both rocky and soft substrates, we see here in the blue box plots that there are only between approximately 12 and 5% of the original native biodiversity that can still be found with living individuals on the Israeli Mediterranean coast. When looking into more detail, we see that those native species that are still there are actually quite small. So the second graph here shows the ratio of the historical maximum body size of the specimens compared with the actual observed body size of recent samples. And we see that the blue ones, so again, the native species, are not even reaching half the maximum body size, which can be regarded as a proxy of reproductive maturity. So this is really dramatic, right? And it's very clear that there is a major turnover going on in these communities. However, what is much more difficult to judge is how this affects the functioning of the ecosystem, because each species has a certain set of biological traits, and the abundance of the species also determines the abundance of these traits being represented in the entire assemblage, which directly governs the ecological processes, for example, energy transfer through the food web or secondary production. And this in turn determines ecosystem goods and services provided and important to society. So our own aim was to gain further insights or new and more detailed insights into the patterns of the Lessepsian invasion and the consequences it has, not only on the native biodiversity, but also on the properties and the functioning of the entire ecosystems. Specifically, we were interested in whether the native species, which are so rare and few today, have been replaced by ecologically similar non-indigenous ones from the Indo-Pacific. So whether there has been a competitive displacement, or if this decline of native biodiversity may be related to this tremendously increasing seawater temperatures, as has been suggested by some authors based on experimental evidence and also on the nature of these losses. On the other hand, of course, knowing whether the original species set is replaced by an ecologically similar one or not also tells you a lot about the potential to impact ecosystem functioning, because if you replace something with something else that is similar, of course, we won't expect high impact, and the other way around. And finally, which is very important also for the study of invasion dynamics, we wanted to know whether the detection of non-indigenous species can be improved in a practical way. Regarding the first question, there are very contrasting or very hot debates going on and contrasting opinions, with some stating that the success of Lessepsian species and the loss of native ones is in fact related to competitive displacement. This is mainly based on evidence concerning some ecologically similar species, and it's also a very reasonable assumption. On the other hand, recent studies on fishes focusing on regional species lists and large-scale macroecological approaches found the contrasting pattern, namely that non-indigenous species which are abundant and successful predominantly settle in empty niches or occupy trade space that is still available. This is in fact very well supported by ecological theory, including very famous concepts such as Darwin's naturalization hypothesis or limiting similarity, which all state that niche differences should be favorable for invaders because they can avoid competition with native species. So the problem is, even if this is well supported by theoretical assumptions, it may still represent a shifted baseline. So we may today see trait differences simply because in the past, functionally similar native species have already disappeared without anyone noticing. And this is also quite reasonable or probable because the Suez Canal was opened already 150 years ago, whereas only several decades later, the first quantitative data sets on soft bottom fauna in the region were available. But the first Red Sea species already occurred a few years after the opening of the canal within the Mediterranean. When it comes to hard substrates or the intertidal, data are even available only since around 2010. So we see a highly altered ecosystem, and it's really hard to judge what has been going on. To overcome the lack of baseline was the aim of the historical ecology of Lessepsian invasion project, conducted at the University of Vienna and conceived and led by Paolo Albano, who is now a researcher at the Zoological Station in Naples. And it was hosted in the Conservation Paleobiology Working Group of Martin Sushin here in Vienna. We had very valuable support in all ways, basically from Israeli researcher, including Yoni Belmeka from Tel Aviv University and Bella Galil from the Steinhardt Museum of Natural History, also in Tel Aviv. Furthermore, Maria Božňák from Zagreb Natural History Museum also supported us a lot. And of course, this would all not have been possible without the help of numerous students and interns. Specifically, the project aimed at exploiting so-called death assemblages to overcome the lack of baselines and reconstruct past communities. The death assemblage is simply speaking the accumulation of taxonomically identifiable skeletal remains of organisms that can be found either in the landscape or in the upper mixed layer of seafloor sediments. This, for example, can be accumulations of mollusk shells, forum tests, ostracods, and the like. The important feature is that all these remains are very durable, and because you have usually low sedimentation rates, death assemblages accumulate over timescales of decades to millennia, so they are time averaged. This has very important implications. One of them is that they tend to accumulate rare species, which are very hard to be found alive or in traditional living censuses because they are so rare or because they inhabit cryptic habitats or are patchily distributed, for example. On the other hand, because so much time is integrated in a death assemblage, they are also compositionally inert, meaning that whereas living communities may change fast, the death assemblage still remains its former state, and it's a very valuable archive of community structure, both taxonomically and functionally. This can be exemplified by, for example, the seagrass meadow, which is a threatened ecosystem in the Mediterranean Sea. Now, if this is, for example, destroyed by some physical impact like boating, anchoring, and other stuff, fisheries, for example, the living community changes a lot, very rapidly. The seagrass is gone, and of course, also all the associated species will no longer be found alive. If you go there, let's say 1 year later, you will find probably a typical, maybe impoverished soft bottom fauna. But if you go and take a sample of sediment and you extract, for example, the molluscan shells, you will still find a high dominance of typical seagrass dwellers, which tells you that in the past, the ecosystem looked differently. With this approach in mind, we went to the Israeli Mediterranean shelf, which is one of the most heavily affected areas by the Lessepsian invasion and native biodiversity loss. And we tried to cover as many different habitats as possible in depth down to 40 meters, including both soft and hard substrates and 2 seasons, and using methods that allow the simultaneous collection of living mollusks and their shells, which of course on the soft bottom you can do, for example, with a graph, which samples both. On hard substrates, it's a bit more difficult, but we used a very nice device called the airlift sampler, which is a kind of vacuum cleaner that can be operated by a diver and used to collect organisms on all kinds of hard substrates. To give you some impressions, what we saw here are, for example, rocky bottoms at different depths, a sandy bottom, and the diver actually working with the suction sampler. Apart from the community data that we could extract from those samples, both alive and dead, we also compiled for each of the species found information on ecologically important functional traits, for example, body size. And in this regard, mollusks also prove a very valuable study system because they are ecologically highly diversified. For example, you have quite large-sized ones, as this Indo-Pacific conch, and also tiny ones, as seen here in the upper right corner, which reach just a millimeter or even less in adult body size. Feeding habit is another trait which is highly important because it determines trophic interactions and energy flow through ecosystems. And we had also information on several others. All this was compiled in a fuzzy-coded trait matrix, and I'm not going to talk about details here, but in short, it's just a way to code trait information in a more natural way than a binary coding, because it allows you to consider, for example, that the species does not only belong to one feeding guild, but it may be very flexible and also have different feeding habits to various degrees. We then aimed at comparing the native and the non-indigenous components of the molluscan assemblages, first within the different stations, using an abundance-weighted trait composition, which is similar to a taxonomic composition, and then also checking whether there are any global patterns that hold across all those highly different habitats. So revealing general differences between the invasive fauna and the native one. And finally, we also assessed the functional diversity of the two faunal components, showing, for example, whether the non-indigenous ones can only perform a subset of the native functions, or maybe the same amount or even more. What we found was quite interesting and also very consistent. These are ordinations of abundance-weighted trait composition, so showing basically the functional profiles of the alien and the native assemblage components within habitats. For example, at this station, which is a soft bottom at 20 meters, as you can see here, there's a very clear segregation of the living assemblages of native species, here shown in blue filled symbols, and the alien living assemblage shown here in red. The death assemblages, so the accumulations of shells, show a very similar segregation, and they are functionally very close to the living assemblage, showing that also historic assemblages were not displaced by the alien ones, because then we would expect them to overlap with the blue symbols here. In some stations, we saw some offset between the native living assemblage and the death assemblage, and the same for the alien ones, but in a parallel way, meaning the similarity didn't change across time, which implies that the factors that cause this offset may equally apply to both assemblages. For example, there may have been an environmental change that shifts trait composition in a similar way for native and non-indigenous assemblages. Of course, this pattern is nice, but to make sure it's not the shifted baseline, we employed some radiocarbon dating of shells, and this is quite hard to read. But if you focus on this area here in the red square, you see that in many stations, time averaging or the median age of the assemblages was very high, and most shells actually predate the opening of the Suez Canal, showing that what we saw is actually a genuine pattern rather than a shifted baseline, which is very important and tells us that the niches of abundant Lessepsian non-indigenous species have always differed and still differ from those of dominant native taxa. When we look at traits individually, the patterns are, of course, very complex, as you may also expect from those very diversified habitats, but they were all significantly different. And for example, here, when we look at body size, you could also see that the non-indigenous species the abundant ones tend to have either smaller or larger sizes than the native ones, which tend to be more represented by medium-sized individuals. Of course, what that means is very hard to predict. And because these assemblages are so dynamic, we also don't know how abundances of species may change in the future. So to get an idea about this, we also calculated functional beta diversity, which basically quantifies the difference in the functional space occupation between the native species and the non-indigenous ones, but independent of abundance. So basically, if you factor this out, you could see that still the beta diversity in both soft and hard substrates was pretty high, especially in the soft ones. But what is even more important is that most of it was related to the turnover component, meaning that they really occupied different trade space, different niches. So when we have an increasing dominance of non-indigenous species because of native diversity decline and collapse and invasion, we can expect that the functional properties have changed dramatically over time for those molluscan assemblages. What you saw here is all about the subtidal, which was heavily affected by the loss of native species. Whereas in the intertidal, such a loss was present, but not as pronounced. Importantly, however, in the intertidal, we have very harsh abiotic conditions and very variable conditions. So this may restrict the number of species that can occur there because they need to cope with these conditions, and also restrict the set of functional traits that can be represented, right? Because certain life strategies are mandatory. For example, being able to withstand desiccation, irradiance, extreme temperatures, and the like. So to have a look at these systems, we chose 4 more stations covering the entire coast of Israel in the Mediterranean Sea, and again sampled mollusks on 1 square meter quadrats. What we found were very impoverished assemblages, or very species poor assemblages. In this case, for example, we had only 8 different mollusks, of them 5 native and 3 non-indigenous species, such as this Indo-Pacific limpet. And as expected by ecological theory, also the trait diversity was very restricted. For example, in 3 out of 5 traits, all the species had just 1 category represented, which was being a hard substrate dweller, having an epifaunal lifestyle, and being non-host associated, so not parasitic or commensal. This may seem quite obvious, but it really shows how restrictive this environment is. So if an invasion occurs there, we may expect that the alien species could have very similar traits to the native one and serve or pose a potential threat. This is also revealed by the function analysis. Again, here abundance-weighted profiles of the non-indigenous species in red and the native assemblages in blue. And the arrow shows that although there is some separation, approximately 50% of trait profiles of the red and blue symbols are actually overlapping, meaning they are identical. This was related to the high dominance of 2 species of limpets in this system, one of them being a native and the other one being an Indo-Pacific species, the one I showed on the image before. And those 2 species had the same trait profile, so they're ecologically very similar, and they drove the entire pattern in these ordinations, as can be seen here on the right. So the more limpets are represented in these symbols, the closer they are in the ordination plot. So we may assume that species that have the same trait occupy very similar niches and may engage in competition. And indeed, we saw that in locations where the native limpet was large, there was an only low prevalence of the non-indigenous limpet Celana. So, and the size differences between, for example, the Ashkelon site and the site in Tel Aviv were quite pronounced. The only difference here was basically the amount of non-indigenous limpets present. Modeling this with a quantile regression approach, also showed that there is a negative correlation between the proportion of non-indigenous limpets, so potential competitors on the left, and the size of the native limpet, both for the median and the 95th percentile. On the other hand, turning the thing around, there was no effect apparently on the size of populations of the Indo-Pacific snail compared in relation to the proportion of native limpets. This, of course, is not causal evidence, so you would need to do really manipulations and experiments to show whether competition is going on or not. But it's a very common phenomenon in intertidal gastropods. And because of the pattern we saw, it's also the most likely explanation here, especially because the genus of non-indigenous limpets involved here is known to be a very aggressive competitor in other localities. So when we take this together, what have we seen so far? We saw the subtidal with its diversified habitats and high species richness, but a dramatic loss of native biodiversity. On the other hand, we saw that in the intertidal, we have species-poor assemblages with potential competitors and co-dominant species, native and non-indigenous ones, overlapping in their niches, but no major biodiversity decline going on. So it seems unlikely that the non-indigenous species are the primary driver of native biodiversity loss. Instead, as proposed already by evidence for some individual species, it seems that the incredibly fast warming of the Eastern Mediterranean may exceed the physiological tolerances of native populations, which are already living at their thermal maximum, because this region was always the hottest part of the Mediterranean. So we always had these edge populations that might be particularly sensitive. When you look at the graph of summer temperature development, it is here a warming since approximately 1980 of about 3 degrees, which is really a lot and much faster than the global average. The intertidal may be less affected because these species are already adapted to extreme and fluctuating temperatures, and they may be less sensitive than the subtidal ones. So with ongoing warming, it doesn't really look very bright for the native species. And on the other hand, the invasion is still going on. When we remember this graph of showing new records on the Israeli coast, it may even be possible that introduction rates are increasing, which may make sense with the continuous enlargement of the Suez Canal on the one hand, and the warming on the other hand, which may make conditions more favorable to those tropical species. However, these trends may be biased, or they are very likely biased, because the detection of non-indigenous species involves often significant time lags, which may be sometimes decades. This is related to the small size of many of those species, so they are difficult to sample, and they will not be readily recognized by citizen scientists or the public. Or they inhabit cryptic or difficult to sample habitats, for example, those rocky reefs with their crevices and the like. Prominent example of detection time lags is the blue-spotted cornet fish, Fistularia commersoni, which is actually quite a large-sized and obvious species, and it may be caught by fishermen, but still, it was possible to backdate a specimen that was not recognized and present in the collection of a fisherman by a quarter of a century. So even those obvious and easy to spot taxa have these time lags. And now imagine how it is with small-sized microgastropods. On the other hand, with the latter, we also face a lot of taxonomic problems. So many groups are quite diverse, but they are pretty featureless, and they need really experts to identify them, and still they are understudied. This means that the taxonomy is not really settled, and if species are newly described from the Eastern Mediterranean, it's always possible that you're either dealing with an overlooked endemic, because the area has received rather little attention in the past, or on the other hand, a newly introduced non-indigenous species that is not known from its native range yet, but first found within the Mediterranean. So it becomes increasingly difficult to identify what is alien and what is native. For example, recently, 2 species of these nice gastropods have been described, and the authors claim that they're either endemic or they could possibly be alien Lessepsian species from the Indo-Pacific or the Red Sea. Just by coincidence, we found shells and specimens of those also from samples in the Red Sea, so we could prove that in fact there are Red Sea species. But this, of course, is a serendipitous finding. As a result of the project, we could report 25 new non-indigenous mollusks for the Eastern Mediterranean, mostly the Israeli coast, 10 more where we were not sure what they are, and new occurrence data for another 15 species. So this shows how much underreported those invaders are and how much farther they may already be distributed within the Mediterranean Sea. So how could we find so many, or what was the reason for this? In fact, it involved, of course, a large sampling effort because of the different habitats covered by the study, the different depths. Chances are high that you find a large diversity of those species. On the other hand, we for the first time sampled hard substrates for small-sized organisms using the airlift sampler, and we used small sieve mesh sizes smaller than usually done in ecological studies to limit the workload. But we found that many of those species are really small and they escape attention if you use larger mesh sizes. On the other hand, we were lucky to have a broad network of collaborators from different institutions all around the Eastern Mediterranean, involving both professionals, but also citizen scientists and private collectors. And finally, which is very important, the conservation paleobiology approach involving the study of death assemblages. This is very underexploited in invasion science, but it has a lot of potential. This is because of both properties of death assemblages. Because they are time averaged, we may find species that are very rare, or maybe even constitute failed invasions. These species belong to clearly Indo-Pacific taxa, although we may not know the species in all cases, and they're only found as empty shells and occur very rarely. It's, for example, these here in the gray square. It's highly unlikely that we would find them in a living census. So the death assemblage already adds quite a lot of new species to any census of potential invaders. On the other hand, because death assemblages are taxonomically inert or compositionally inert, it's possible to find species that are alive only and suggests that these are new arrivals to the system. Because they are only very shortly present there, they may not have contributed enough shells to leave a significant signal or a detectable signal in the death assemblage. So if you find species alive only, it's a good sign that they may actually be new to the system, either range expansions or in this case, most likely of Indo-Pacific origin. So this was actually the results of the historical ecology project. But of course, there's much more work to be done. So we started a new project, which is dedicated on the drivers of native biodiversity loss in the region. And we were lucky to be allowed to use the Israel National Benthic monitoring data, which covers a set of 13 stations all along the coast of Israel and constituting soft bottoms at comparable water depths. And Aaron, I would like to ask you now to maybe stop the recording for the next 2 slides. So taken together, we've seen that in the subtidal realm of the Eastern Mediterranean, invaders have different traits and occupy preferentially different niches than the native ones. Whereas niche overlap occurs in the intertidal habitats where trait diversity is restricted by the environmental boundary conditions. This means there's no evidence based on this data for a large-scale displacement, regional scale one, of the native species by the invaders, which may sound good. I mean, but still, it highlights on the other hand that another driver, the human-mediated warming, is in fact the major culprit that drives native species to extinction on the one hand. And on the other hand, if we have functionally different invasive species, it means that these have a very high potential to change the functioning of the entire ecosystem with highly unpredictable impacts for the services and also the resilience. So it's really a major human-driven change in the system that is caused by warming and, of course, the presence of this highly potent vector, which is the Suez Canal. The results of the project also show that the extended temporal perspective that can be obtained by the conservation paleobiology approach and the use of natural archives of community composition can help to discriminate competing ecological scenarios in cases where observational baselines are not available, which is frequently the case, as we've seen with the old invasions, which are actually much more common than was previously appreciated. The analysis of death assemblages can also help to detect non-indigenous species faster than focusing only on the living individuals, and in taxonomically difficult groups, the presence of live-only specimens may also help to develop a preliminary hypothesis, at least, of their potential native or non-indigenous status. So overall, the integration of approaches from paleontology and ecology can advance the understanding of biological invasions, not only in this system, but also in others, and help us to judge better their impact, although, of course, much work is still to be done. And with this, I would like to end here and thank all the colleagues who provided invaluable help and made all this possible, and also to the Israel Nature and Parks Authority, who granted permits for sampling and their support. And of course, I would like to thank you also for your attention. Thanks a lot.
Thank you, Jan. Sorry, everyone, Aaron had to leave because he had an emergency at home, so he left me. We have one question in the Q&A. Is there any evidence from sclerochronology over the growth rate and age at death of the now small indigenous species? Has one or both reduced?
No, we have not looked into sclerochronology because most of the species we are dealing with are really tiny microgastropods, and those are actually very short-lived usually. So they have life cycles that are maybe just a year or even less sometimes. And we just found that the specimens that represented the native species were really small, and they tended to appear only in spring. So we had post-metamorphic specimens or very juvenile specimens in our spring sampling campaigns, but almost all of those had disappeared in summer, which also suggests that there's some recruiting going on from healthy populations which may live in deeper water or cooler waters, or in other geographic locations, because some have long-lived planktonic stages. But yeah, this would certainly be a very interesting topic for larger sized species. The problem is that these are basically not available anymore. So it's extremely difficult to find them.
Thank you. So are there any other questions? So Giacomo is asking, thank you for your excellent talk. In your sediment samples, do you ever find fish otoliths? Yeah, I can answer that.
I guess you know the answer. Yes, there were quite a lot. And this is nice also because the Israeli shelf is an open shelf, and there are very low there's very low sedimentation, so the time averaging is quite good, and we can often find a good number of otoliths also in the samples, especially from the soft substrates, which are now being studied by Konstantina. And they already actually provided some very important results that have been published.
So does anyone else have a question? You can also raise your hand, or if you prefer to write. Okay, so I will stop recording. Okay, one more question. Andy is asking, are there any species that have gone the other way? Yeah.
Yeah, this is a long debate, in fact, and there have been claims of several authors that there are species that are anti-Lessepsian. I'm not a specialist for all taxa, of course, but for the mollusks, there have. But this has all turned out to be not really well supported. So people looking for those did not really find any populations of Mediterranean species going to the Red Sea. Sometimes there were archaeological findings of shells that were mistaken. And there are also some taxa that are incredibly similar between the Mediterranean Sea and the Red Sea. So they are closely related and they almost look the same. And it's very easy to confuse them. And we have some species that apparently occur on both sides, but I highly suspect that these are cases of those very similar twin species, but in fact, different taxonomic entities. And of course, another factor that may be problematic for Mediterranean species is that the Eastern Mediterranean is very warm and it represents already a stressful environment to those native populations. And the Red Sea, though not the Gulf of Suez, but other parts are even warmer than that. And maybe, I mean, it seems very unlikely that they could establish there.
Okay, thank you. So if there are no more questions, then I would like to thank you all. Thank you, Jan.
Thanks a
for this
lot.
very nice talk. And Aaron asked me to remind you that our next talk of the seminar series would be in 7th of December by Rowan Lockwood, who would talk about the historical ecology of oysters in Chesapeake Bay. Thank you very much, and looking forward to the next one.
Bye.